Hi everyone, my name is Azila Adnan from University Malaysia Terengganu, Malaysia. So I would like to share my presentation today entitled Bacterial Cellulose or BC and Emerging Future Biomaterial. So the sources of cellulose actually we can get our cellulose from plant and also from the bacteria. However, from plant specifically, we can get our cellulose from the cell walls here. And then from the bacterial cellulose, we can obtain our cellulose through a fermentation process by some kind of bacteria which is called as Acetobacter bacteria that secretes cellulose nanofibers. So if we compare the plant cellulose and bacterial cellulose, there are some advantages of BC over plant cellulose that will include it is finer and more complex. There's no hemicellulose or lignin or pectin that present in bacterial cellulose. The fiber is much stronger and it can be grown to any shape and thickness that we desire. It can be produced on a variety of carbon source or sugar source and also nitrogen source. So carbon source, we could actually use glucose, sucrose, fructose, maltose, lactose, or glycerol, or mannitol, or sorbitol. So nitrogen source, we could use the well-known one, which is yeast extract, beef extract, soybean extract, corn stick liquor, and so forth. And in terms of biocompatibility and biodegradability, if we compare with these two types of cellulose, bacterial cellulose has higher degree of biocompatibility and biodegradability. As an overview, in 1838, the cellulose was actually first discovered by Anselme Payen, and he was able to actually isolate the cellulose from plant and then he characterized it. And by the way, in 1886, the A.J. Brown discovered the cellulose that has been synthesized by a kind of bacteria and specifically from Acetobacter xylinum, and then it was called as bacterial cellulose. So bacterial cellulose is actually a homopolysaccharide that has glucose monomer or glucose repeating unit that combines by a bond that we call as glycosidic bond. And biocellulose or the bacterial cellulose is actually non-toxic, non-carcinogenic, and really high biocompatible material. And the porous structure of BC is really good for a physical barrier against external infection. So that BC is well or widely used in wound healing biomaterial application. And also BC has high water holding capacity or water retention ability due to the hydrophilic behavior. So that will help the wound heals better and faster when the wound is being constantly moisturized. And again, bacterial cellulose is free from hemicellulose, pectin, and lignin. So these are the look of the BC pellicles. So the A diagram is showing the BC pellets, while B and C are showing the sheet or the layers of bacterial cellulose membranes. Okay, there are types of application of BC as a versatile biopolymer. So the first one we can see here, it is widely used in biomedical industry or application. So usually bacterial cellulose is used to make artificial blood vessel and also artificial skin, which is applicable to the skin burn or skin burn patient, and also it can act as a wound patch for wound healing mechanism, and also it is quite attractive in drug delivery application, and also making audio devices, and also in textile and paper industry. So for textile and paper industry, bacterial cellulose is starting to to gain an attraction or attention from the researchers to make biocellulose clothes and also nanocellulose papers. And meanwhile, in food industry, we already know that biocellulose or the bacterial cellulose is really useful in making nata de coco, the famous Filipino dessert, and also it can be used as a fat replacer. So these are some findings that has been obtained. So if we look at this picture, these are the structures of cellulose nanofibrils from under the scanning electron microscope. And so for A, B, C, D, E, F, the bacterial cellulose was actually fermented under different carbon sources. So if we can see that A was fermented in glycerol media, while B in sorbitol media, C mannitol, another alcohol sugar, and fructose is another monosaccharide, and E in disaccharide, which was glucose, and also F was in glucose media. So if we compare the structures, we can see that the different types of carbon sources or sugars affect bacterial cellulose appearance under the scanning electron microscope. So for agitation speed during the fermentation process is actually affecting the BC production as well. So we know that agitation speed or the mixing speed during the fermentation is crucial or important for mixing the media to supply even air or oxygen in the reactor. However, if we increase the speed beyond 80 RPM, roughly here, we can see that the BC production or BC yield was decreasing. So perhaps it could be due to the loss of substrate by direct oxygen and also due to the shear force during the agitation process. So another one is we applied additive such as riboflavin, vitamin C or ascorbic acid, and also ethanol. And the final one was vinegar or acetic acid. So we can conclude here, we can see that different types of additive affected BC production or BC yield. And if we have a close look, ascorbic acid or vitamin C produce higher BC yield or BC production compared to riboflavin, ethanol, and also acetic acid. So if we look again, the riboflavin here shown to decrease the BC productivity. That was in agreement with a study by Ishikawa in 1995 with his co-workers. And also we can see that alcohol or ethanol shown to inhibit BC production at higher concentration at 1% volume per volume. So that is also agreeing the study by Zhu et al. In 2022. Meanwhile, the acetic acid shown to have no obvious effect on the production of BC, which is also supported in a study by Aswini and co-workers 2022. So another one here I would like to share was if we ferment the bacterial cellulose or produce bacterial cellulose under two types of Hestrine Schramm media, which the green one is the conventional or the common Hestrine Schramm media, and also the yellow bar here is showing the modified Hestrine Schramm media. So the green one shows lower production of BC yield. However, in modified HS media or Hestrine Schramm media, we can see the BC yield is higher. So the modified Hestrine Schramm media was containing glycerol constip liquor that proves to produce more BC or bacterial cellulose compared to glucose consistent liquor as their carbon and nitrogen source. So we can conclude that glycerol is actually can allow a consistent production throughout the growing time and in larger amounts of BC. Okay, so for a conclusion, we can see from the earlier bacterial cellulose, which is the emerging biomaterial and also biopolymer, has numerous applications such as biomedical, food industry, textile and paper, drug delivery, and also other types of application as well, including the audio device making. So in future, we can say that it takes a multidisciplinary approach to consider the feasibility and also the potential benefits of BC in the development of other applications that might benefit the humankind.